Hello, today we're going to discuss bacteria. So, prokaryotes. There's mainly two different kinds of prokaryotes, is bacteria and archaea. Bacteria belong to the domain bacteria. They exist in nearly every environment on Earth. They're important to the human body, industry, and food production. Archaea belong to domain archaea. Archaea tolerate extreme environments. They have similar proteins to eukaryotic cells. They're mostly anaerobic, and they include halophiles, which is salt-loving, methanogens, use of CO2 and give off methane. And then there's thermoacidophiles, which are high temperature, low pH. Additional differences. So bacteria and archaea have differences in cell wall proteins, lipids in the plasma membrane, and ribosomal proteins and RNA. Those are the main differences. If you want to read the additional differences, you can see that in that chart on the left. Okay, structure of prokaryotes. They have some characteristics of all cells, such as DNA and ribosomes. They have a long circular chromosome found in the nucleoid. So the nucleoid is the central region in a prokaryotic cell that contains the chromosomes and has no surrounding membrane. So it's like a nucleus, but there's no actual structure. They usually have at least one smaller piece of DNA called a plasmid, which is also circular. So the plasmid, a genetic structure in a cell that can replicate independently of the chromosomes, typically a small circular strand in the cytoplasm of a bacterium or protozoan. Okay, more about the structure of prokaryotes. Some prokaryotes secrete a layer of polysaccharides around the cell wall, forming the capsule, which prevents cell dehydration and helps with attachment to surfaces, protects from antibiotics. Pili are hair-like structures made of protein. Pili help cells attach to surfaces and serve as bridges between cells that send plasmids to each other. Prokaryotes are only 1 to 10 micrometers long and about 0.7 to 1.5 micrometers wide. Their small size makes nutrient diffusion easy due to their high surface area to volume ratio. Okay, structure of prokaryotes continue. Some prokaryotes are stationary, but others use flagella for movement. Prokaryotic flagella are made of filaments, unlike flagella of eukaryotes, which are made of microtubules. Other prokaryotes glide over a layer of secreted slime. There are 3 different shapes of prokaryotes. There are bacilli, which is rod-shaped, cocci, which is round-shaped, and spirilla, which is spiral-shaped. And you can see each of those 3 differences on the left. Okay, Gram staining. So scientists classify bacteria based on the composition of their cell walls. All bacterial cells have peptidoglycan in their cell walls. Gram staining is a common procedure for identifying main kinds of bacteria. Bacteria with large amounts of peptidoglycan appear purple when stained, which is gram-positive if they have that. Bacteria with lipid bilayers have less peptidoglycan and appear pink when stained. Those are gram-negative. So if there's a lot of purple when stained, that is gram-positive, not a lot gram-negative. Again, the positive means there's a lot of this peptidoglycan in their cell walls. Depending on whether a bacteria is gram-positive or gram-negative, that's going to determine the best kind of antibiotic to treat the bacterial infection. Okay, Bacteria replicate by the process of binary fission, so they don't have a nucleus, so they don't undergo the process of mitosis. Instead, they undergo binary fission. So in biology, binary fission is a type of asexual reproduction where a parent cell divides, resulting in two identical cells, each having the potential to grow to the size of the original cell. Okay, more about prokaryotes. So most prokaryotes are classified as a kind of autotroph. So an autotroph is an organism that is able to form nutritional organic substances from simple inorganic substances such as carbon dioxide. Essentially, autotrophs are capable of making their own food. If we look at this little mini cladogram here, there's photosynthetic autotrophs, which are autotrophs that use basically light, or photo, that's the root word there, to make their own food. Chemoautotrophs use chemicals to make their own food. Then at the other end, there are heterotrophs, which is an organism deriving its nutritional requirements from complex organic substances. Essentially, heterotrophs are organisms that have to consume other organisms to gain nutrients and sustenance. Okay, bacterial survival. So, with mutations, genetic mutations can help bacteria survive in changing environments. Mutations allow for genetic diversity in an asexually reproducing population. Not much, but could potentially be enough for survival. So really the only mutations they're getting is from potential exposure, but really it's more from small errors in the replication process. Some mutations can lead to changes such as antibiotic resistance. With endospores, so endospores are dormant cells produced in response to harsh environmental conditions. So poor conditions, so the spore coat surrounds a copy of the chromosome in a small part of the cytoplasm, and then in favorable conditions, the spore germinates and grows into a new bacterial cell. So basically, endospores can happen very quickly or over a long period of time to basically survive, however long it takes to survive this harsh environmental condition. And you can see a diagram of this whole endospore germination process on the right. Okay, pros and cons of bacteria. So pros, bacteria are decomposers returning vital nutrients to the environment. Some soil bacteria fix nitrogen, which is vital for amino acid, DNA, and RNA synthesis. Some nitrogen-fixing bacteria live in symbiotic relationships with plants and crops. Your body is covered in bacteria inside and out. Normal flora have a symbiotic relationship with humans. They prevent disease, aid with digestion, and make vitamins. Bacteria are responsible for some food production, commercial production of vitamins, and they can be used to fight disease and produce antibiotics. Cons of bacteria: a small percentage of bacteria do cause disease. Bacteria cause disease in two ways. They multiply quickly at the site of infection before the immune system responds, or they secrete a toxin or harmful substance.
Thank you for watching. Please share and subscribe, and remember to spread